Now, in pathology, we study the disease process, about the structural changes, biochemical changes, which will lead to the clinical manifestations. So basically, there are four aspects of disease process. First of all, what is the etiology of the disease? What is the initiating cause of the disease? Which can be broadly divided into two, either the genetic causes, which can be either inherited mutations or acquired mutations, or it can be environmental, that is related to nutritional, infectious disease, etc. The next aspect is the pathogenesis. In pathogenesis, we study the mechanism of the disease. Now, what is the structural alteration, functional alteration, biochemical alteration that is leading to the disease? And nowadays, more and more research is coming up with the development of the omics, like the proteomics, genomics, so that we can learn about the pathogenesis of the disease, so that more and more biomarkers can be developed, so that we can learn about the progression of a disease. Next is, because of the pathogenesis, what are the morphological changes that are happening in a cell? Basically, this is what we see in pathology under the microscope, the morphologic changes, and to give a diagnosis of the disease process. And nowadays, it is not only limited to the diagnosis of a disease, but also we see whether we can find any immune markers through the detection of protein expression and immunohistochemistry. Like in case of breast cancer, we detect the HER2 new expression so that we can predict whether the drug trastuzumab will act or not. Similarly, we have the PD1, PDL1 immunoexpression markers so that we can say whether the immunomodulators will act on cancer cell or not. Now, the final thing what we see are the clinical manifestations because of all this process. What are the signs and symptoms because of this pathogenesis and morphologic changes, and what will be the disease outcome? Now, virtually all diseases start with the molecular or structural alterations in cells. Now, this concept of the cellular basis of disease was first put forth by Rudolf Virchow, who is known as the father of modern pathology. Now, Virchow emphasized the idea that the individuals are sick because their cells are sick. So now we are coming to today's topic, that is how the cells become sick. Now, normal cell is in a state of homeostasis. The normal cell is subjected to several stress. However, it tries to maintain a balance, and that is called as the homeostasis. One should not confuse this term with the term hemostasis. Hemostasis is stoppage of blood flow, which you will study in the chapter of hemodynamics. Now, when a normal cell is subjected to stress because of, say, ischemia, decreased oxygen supply, or whatever stress, first of all, it will try to adapt to that stress. Now, what is adaptation? That is a reversible functional and structural response to the changes in the physiological states or some pathological stimuli. Physiologic states like pregnancy, in which the uterus enlarges. So, in adaptation also, we have four basic adaptations, that is hypertrophy, that is increase in the cell size, hyperplasia, increase in the number of cells, atrophy, like in case of disease atrophy, suppose you have put in a plaster, if you are not immobilized for several days, there will be atrophy of the muscle. So this is also a type of adaptation. And the last adaptation is metaplasia, in which one type of epithelium is changed into a different type of epithelium. Like in case of chronic smokers, we can see that the columnar epithelium of the airways changes to the squamous epithelium. So this is called as squamous metaplasia. Now, when the cell is unable to adapt and the injurious stimulus is continuing, the cell will be injured. Now, if the injurious stimulus is mild or transient, then the sort of injury that will be a reversible injury, means the cell can again go back to its normal state. However, if the cell injury is severe and progressive, like if the oxygen supply is cut off for a long time, then the cell will go into a state of irreversible injury, and ultimately the cell will die. And there are basically two types of cell death. One is necrosis, and another is apoptosis. There are other features of cell death also that I'll discuss in the next class. Now, besides that, there can be other morphological changes also, like intracellular accumulation of substances like lipids, proteins, etc., and calcification. So, prototype example, say we have a normal myocyte, that is heart muscle, which is subjected to hyperdynamic circulation because of hypertension or say anemia. So, to deal with the increased load, the cell will try to adapt and the cell size will increase because the heart muscles cannot divide. So, there will be no hyperplasia, there will be only hypertrophy. However, if the hyperdynamic insult continues, then the cell, first of all, there will be reversible sort of injury. And if the insult continues, then ultimately there will be cell death. Now I'll deal with the causes of cell injury. That is what is the etiology. So first and foremost, the most important thing is oxygen. We survive on oxygen, all our cells survive on oxygen. So oxygen deprivation, hypoxia causes cell injury by obviously reducing the aerobic oxidative respiration. Now what can be the causes of hypoxia? It will be either reduced blood flow, which is called as the ischemia, inadequate oxygenation of blood due to cardiorespiratory failure, means the blood flow is adequate, but because of the deficiency of oxygen, because of respiratory failure, there will be inadequate oxygenation. Decreased oxygen carrying capacity of blood, as we can see in anemia or carbon monoxide poisoning, or after severe blood loss. Now, what are the other causes? There can be physical agents like mechanical trauma, extremes of temperature, sudden changes in atmospheric pressure, radiation, electric shock. Chemical agents and drugs, simple chemicals like glucose or salt in hypertonic concentration, oxygen at high concentration, poisons like arsenic, cyanide, mercuric salts, environmental and air pollutants, insecticides, herbicides, occupational hazards like carbon monoxide and asbestos exposure, or even drugs like alcohol, or the increased use of the therapeutic drugs can also be acting as an injurious chemical stimulus. Besides that, there can be infectious agents which can range from submicroscopic viruses to large tapeworms, and in between there can be bacteria, fungi, or higher forms of parasites. And then there can be immunologic reactions. We know that the immune system helps us in defense against infectious pathogens, but sometimes these immune reactions can also be hazardous when there is hyperimmune reaction, like in also in autoimmune reaction when the body's immune system reacts against the self antigens. And there can be genetic derangements, like due to deficiency of functional proteins, like the enzyme defects. We have the inborn errors of metabolism, like the Gaucher disease, Niemann-Pick disease, etc. The accumulation of damaged DNA or misfolded proteins that can also trigger cell death, which can be beyond repair, or the variation in the genetic makeup also makes one susceptible to the injurious stimulus. Then there can be nutritional imbalance like protein calorie deficiencies, then deficiency of vitamins, the psychological disorder of anorexia nervosa, that is self-induced starvation, excess of cholesterol that can predispose to atherosclerosis, and then obesity, that is also an increasing disease nowadays because of diabetes and cancer. In addition, the problems of undernutrition and overnutrition is also prevailing. Now coming to the next thing, that is the mechanism of cell injury. Now the cellular response to the injurious stimulus, this will depend on the nature of injury, duration, and severity. For example, if the oxygen supply is cut off for a shorter duration, that may cause a reversible injury. On the other hand, if the process is longer, then it will lead to an irreversible injury. Also, the consequences of cell injury will depend upon the type, state, and adaptability of a injured cell. Like a cardiac myocyte cannot withstand oxygen deficiency for a long time. On the other hand, a skeletal muscle of the skeletal system that can withstand that oxygen deficiency for quite a long. Now, coming to what are the basic main targets of this cell injury. First and foremost, the most important is mitochondria. Next, cell membrane. Then the machinery involved in protein synthesis and DNA. Now come one by one. Now, this mitochondria that can be damaged, how it can be damaged? By increase of the cytosolic calcium, the reactive oxygen species, oxygen deprivation, hypoxia and toxins, or mutation in the mitochondrial genes. The mitochondrial DNA we know is maternally inherited, and the basic manifestations are that of neuromuscular system. So when there is deficiency of oxygen supply or injury due to toxin or radiation, what will happen? There will be decrease in ATP generation, and also there will be increased production of reactive oxygen species due to incomplete oxidative phosphorylation. So this will lead to cell necrosis. On the other hand, also when there is decrease in the survival signals, the pro-apoptotic genes will be activated. Now, apoptosis is also a type of cell death. So this leads to leakage, development of some membrane pores in the mitochondrial membrane, which leads to leakage of the mitochondrial proteins, and this will lead to apoptosis. Now, what are the manifestations of depletion of ATP? Now, ATP we know is produced basically in two ways: the oxidative phosphorylation of adenosine diphosphate. Now, this is the most important way in which ATP is generated. Most of the ATP is generated in this way. The other is the glycolytic pathway. Now, what are the major causes of ATP depletion? One is the reduced supply of oxygen and nutrients, mitochondrial damage as the ATP is generated from mitochondria, and some toxins can also lead to ATP depletion. And depletion of ATP to 5 to 10% of normal levels, that has got widespread effect on many critical systems. Now, what will happen with depleted ATP? We know ATP is important for the function of sodium potassium ATPase pump. Now, when the pump is deranged, the normal function of this pump is to drive out sodium. So, if the pump is deranged, there will be increase of the intracellular sodium and decreased potassium. Now, this increased amount of sodium that will drive water into the cell. The water will move because of osmosis. So this will lead to cell swelling as well as endoplasmic reticulum dilatation. Now, what are the other effects? Now there is alteration of the cellular energy metabolism. So there will be increased AMP because ATP is not generated. So this will in turn stimulate phosphorylase and phosphofructokinase activities, and there will be increased glycogenolysis and glycolysis so that ATP can be generated from anaerobic glycolysis. So this will in turn lead to lactic acidosis, that is increased pH of the cell, that is acidic environment, sorry, decreased pH, that is acidic environment of the cell. So this will in turn decrease the activity of various cytosolic enzymes. Next, the third thing is there will be structural disruption of the protein synthetic apparatus. So this will lead to detachment of ribosomes from the rough endoplasmic reticulum and dissociation of polysomes. So this will ultimately lead to reduction in protein synthesis. Next, after the mitochondrial damage, the next is how the membrane is damaged. Now, membrane damage is integral part of almost all types of cell injury except apoptosis, where the cell membrane usually remains intact. Now, apart from that, it can also be damaged directly by bacterial toxins, viral proteins, lytic complement components, and physical and chemical agents. Now, what are the effects of membrane damage? Now, again, when there is decreased oxygen supply or increased amount of cytosolic calcium, the reactive oxygen species, this will lead to lipid peroxidation. Decreased ATP will also lead to decreased phospholipid synthesis, and we know that the phospholipids and proteins are integral part of the cytoskeletal system. So when these are lost, there will be membrane damage. Also, increased amount of cytosolic calcium, this will lead to phospholipase activation. This will cause degradation of the phospholipid. So there is decreased synthesis as well as increased degradation. So ultimately this will lead to lipid breakdown, and the lipid breakdown products that are also like the free fatty acids or the acyl carnitine, that are also harmful for the membrane. Also, this protease activation that will cause cytoskeletal damage. Now, cytoskeletal proteins helps in anchoring the cytoplasmic membrane to the interior of the cell. So when the cytoskeletal proteins are damaged, there will be breakdown of the cytoskeletal system. Now, what are the manifestations of different membrane damage? When there's mitochondrial membrane damage, we have seen there's opening of the mitochondrial permeability transition pore. So this will again lead to decreased ATP, and there will be a generation of pro-apoptotic molecules, which will lead to apoptotic cell death. The plasma membrane damage, that will lead to loss of osmotic balance and influx of fluids, because of which there will be cell swelling. Also, there will be leakage of metabolites like the glycolytic intermediates, which are essential for ATP. So there will be again decreased ATP. And when the lysosomal membranes are damaged, so this will lead to leakage of the enzymes into the cytoplasm. And when these enzymes are leaked into the cytoplasm, like the RNAs, DNases, proteases, or phosphatases, they will damage the RNA, DNA, proteins, which will ultimately lead to the damage of the cell. Now, next is damage to DNA. We have seen the mitochondrial damage, the membrane damage, next the damage to DNA. Now, DNA damage that can be caused by exposure to radiation, the chemotherapeutic drugs, and reactive oxygen species, or it may occur spontaneously as a part of aging. Now, in a normal cell cycle, we know that there are 4 phases: G1, S, G2, and M. There are 2 checkpoints between G1S and G2 and M. Now, suppose if the cell is damaged or has acquired some mutations in the G1 phase, the purpose of the G1S checkpoint is to check for whether there is any damage, so that this can be rectified before it goes into cell replication. So now P53 is a very important gene, which is called the guardian genome, and this protein or this gene is located in the nucleus. So this helps in detecting whether there is any damage or not. So if the P53 genome finds that there is any damage, the cell, it can ask the cell to go for a complete senescence. That is, it will not receive any growth signal. Or what else it can do? It can cause a G1 arrest. It can cause the cells to be arrested in the G1 phase, and then the cell will be repaired, and then the cell cycle will move on again. Oxidative stress. Now, what is oxidative stress? In oxidative stress, there is accumulation of oxygen derived free radicals. Now, in a stable atoms, we can see that there are paired electrons. Now, what happens in free radical? In free radical, there's the presence of unpaired electron, because of which the radical becomes very angry and there is damage to the cell. Now, and the purpose of the antioxidant is to give an electron to the free radical, so that it becomes stable once again. Now, how the free radicals are formed? It can be formed from ultraviolet radiation, from air pollution, from ionizing radiation, smoking, from different sort of metabolism, and also from inflammatory process. Now, what happens when there is increase? These reactive oxygen species are produced normally in cells during mitochondrial respiration and energy generation, because the reactive oxygen species are not always harmful. They are useful for removing the microbes from if there is a microbial insult, the reactive oxygen species help in removing the microbial insults. And also the reactive oxygen species like nitric oxide, they help in vasodilatation. So it is not always harmful. Suppose if there is increased production, and also the body at the same time has antioxidants to circumvent this effect of increased reactive oxygen species. If there is increased production of reactive oxygen species, as well as there is decreased degradation of this reactive oxygen species, this will lead to oxidative stress. Now, what are the role of free radicals in biological system? Now, the reactive oxygen species can be associated with almost any disease you can think of. Almost all the systems are involved, like in brain, the Alzheimer's disease, which is again a protein misfolding disorder, or in Parkinson's disease, multiple sclerosis, in heart, in kidney, in skin, joints, immune system, lung, eyes, or blood vessels. Any organ you can think, there's a the pathogenesis of reactive oxygen species is there. But it is not always harmful. The positive effects of free radicals are that the presence of low concentration of free radical is important for the normal cellular redox states and immune function. Free radicals are used by phagocytic cells like neutrophils and macrophages to kill the bacteria during infections. And it can also modify the oxidation reduction states. Now, nitric oxide, as I stated, it helps to regulate the blood pressure. Now, how the free radicals are generated. The ones which are marked in red and circled in red, these are your reactive oxygen species. So during a normal oxygen metabolism or enzymatic pathway, this reactive metabolites can be generated like the superoxide anion, and from that superoxide anion, this hydrogen peroxide can be generated. Now, nitric oxide that is normally present in macrophages and endothelial cells, this can react with the superoxide anion and can give rise to a peroxynitrate. Now, certain metals like copper and iron, this can also generate hydroxyl radical, which can act as a reactive oxygen species. So the generation of free radicals, the reduction oxidation reactions that occur during normal metabolic process that can generate reactive oxygen species. The absorption of radiant energy like ultraviolet light, X-ray, that can generate reactive oxygen species. During inflammation in activated leukocytes by NADPH oxidase, the reactive oxygen species are generated to circumvent the microbiological insult. But if it is excess, it can again lead to excess generation of free radicals. Now, if there is complete absence of free radicals, then there can be a genetic disease which is called as the chronic granulomatous disease, where there is impairment of generation of reactive oxygen species, and we see a macrophage aggregate. That's why it is called a chronic granulomatous disease. As well as some intracellular oxidases like xanthine oxidase can also generate superoxide anion. Now, apart from that, enzymatic metabolism of exogenous chemicals and drugs like carbon tetrachloride can generate reactive oxygen species. Now, transition metals, as I said, like iron and copper, this can also generate free radicals. Now, this Fenton reaction is important for you to remember. In our body, the iron is normally present in the ferric state. So, first of all, this ferric state is converted to the ferrous iron, and this ferrous then reacts with hydrogen peroxide, and this generates hydroxyl radical. So this is called as the Fenton reaction. Now, these free radicals are generated. Now, the body also knows how to circumvent this free radical. So, it has antioxidants. So, antioxidants will either block the initiation of free radical or inactivate them. That is, they will scavenge the free radicals. So, what are the normal antioxidants present, like vitamin A, vitamin E, ascorbic acid, and the glutathione in cytosol? And the levels of the reactive metals like copper and iron, that can also be minimized by the help of transport proteins. Like iron is carried by the transferrin so that they are not free in circulation. Copper is carried by ceruloplasmin. Now, there's also an enzymatic free radical scavenging systems. The important enzymes are catalase, superoxide dismutase, and glutathione peroxidase. Now, catalase decomposes hydrogen peroxide. Superoxide dismutase converts the superoxide into hydrogen peroxide, and this is again acted upon by catalase, which is converts it into water and oxygen. So there are two sorts of superoxide dismutase. One is present in the mitochondria, that is the manganese superoxide dismutase. Another is present in the cytosol, which is the copper zinc superoxide dismutase. Now, another important antioxidant system is the glutathione peroxidase. Now, it converts the hydrogen peroxide into water. And the intracellular ratio of oxidized glutathione to reduced glutathione, that is a reflection of the oxidative stress. Now, in the normal body, the amount of reduced glutathione should be more than oxidized glutathione. Increased ratio in favor of oxidized glutathione, that can be seen in disease process like Parkinson's disease, etc. Now, what are the effects of this reactive oxygen species? There can be a lipid peroxidation, which will lead to membrane damage. There will be protein modifications which will lead to breakdown and protein misfolding, which will accumulate and will lead to various disease processes. And also there will be DNA damage in the form of mutations. There can be single strand break, double strand break, which will lead to a mutated form of DNA. Next is disturbance in the calcium homeostasis. Now, calcium normally serves as second messenger in several signaling pathways. Other second messengers we have like cycling AMP, then we have the DAG, inositol phosphate, etc. Now, normally it is present in cytoplasm in very low concentration. The cytosolic free calcium, it is present only in like in 0.1 micromole as compared to the extracellular levels, and whatever intracellular calcium is present, that is present mainly in the mitochondria and the endoplasmic reticulum. Now, when there is any injurious stimulus, there will be increased cytosolic calcium because of release from the mitochondria or say endoplasmic reticulum. And this will lead to activation of cellular enzymes like phospholipase, protease, which will ultimately lead to damage of the membrane by disruption of the cytoskeletal membrane system. There will be activation of the endonuclease, which will lead to nuclear damage, and also there will be activation of ATPase, which will lead to decrease ATP. And also the increased amount of calcium in mitochondria, that will lead to increase in the mitochondrial permeability. That will lead to development of the pore formation. Again, there will be decreased ATP. Now, next is endoplasmic reticulum stress, the unfolded protein response. Now, whenever there is any degenerated protein, or say the protein which is damaged due to ultraviolet radiation, heat, or reactive oxygen species, now this damaged protein is removed by the ubiquitin protease system. The ubiquitin protease system presents this damaged protein or denatured protein to the proteasome, and ultimately this is fragmented. This happens in the normal cell. Now, if there is and this protein folding is done by the chaperone proteins, which helps in the folding of the proteins. Now, if there are some metabolic alterations, genetic alterations, that can lead to excess of misfolded proteins. The system of removal of this misfolded protein is not adequate. This will lead to excess of this misfolded protein. Again, this will lead to cell apoptosis. Now, these are some examples of misfolded protein disorders like the Alzheimer's disease, where there is deposition of the A beta type of peptide protein. In the CJD, that is the prion diseases, we can see the deposition of the prion proteins. Similarly, there is the alpha-1 antitrypsin deficiency, cystic fibrosis, etc. Now, what are the key concepts of this cell injury? There is ATP depletion. There will be mitochondrial damage, which will lead to ultimately to ATP depletion. There is increased permeability of the cellular membranes. There is accumulation of damaged DNA or misfolded proteins, accumulation of reactive oxygen species. There will be influx of calcium, which will lead to activation of several enzymes, which will in turn damage the cellular components, and there will be excess of unfolded protein response. So this will all lead to this cell injury, and these are the mechanisms of how the cell is suffering from injury. Now, after the mechanism, we will come to the morphological aspect, the types of cell injury. Now, normal cell, you can compare to a sailing ship, everything is fine. Now, in case of reversible injury, the people are trying to save the ship, the sunken ship, and it might be still reversible. And in case of irreversible injury, nothing is possible. The ship has already sunken. So first of all, coming to the reversible cell injury. Reversible cell injury, that is characterized by functional and structural alterations, that is in an early stage or mild form of injury, and that can be correctable if the damaging stimulus is removed. Now, what are the alterations seen in reversible injury? There will be generalized swelling of the cell and organelles because of the damage of the sodium-potassium pump and increased influx of water. So this cell swelling will ultimately lead to blebbing of the plasma membrane. There will be detachment of ribosomes from the endoplasmic reticulum because of the swelling of the endoplasmic reticulum, clumping of the nuclear chromatin, and the organs which are rich in lipid, that will show fatty change. So these are the morphological aspects of reversible injury. Coming to them one by one. Now, why there is nuclear clumping? Because of intracellular lactic acidosis. As I had said, when there is decreased oxygen supply, there will be failure of aerobic respiration and there will be shift towards anaerobic respiration, which will lead to accumulation of lactic acid. So there will be decrease in the intracellular pH and there will be, so this is the reason of clumping of nuclear chromatin. Now, this is a normal tubular cell of a kidney. We can see this is the normal nucleus, while this nucleus is appearing more clumped. So this is called the clumping of the nuclear chromatin. Now coming to damage to the plasma membrane, what it will lead to? This will lead to the hydropic swelling and other membrane changes. So this lack of ATP, which will obviously affect the sodium potassium pump, and this will lead to influx of water, and because of this we will see this hydropic swelling. This is again a kidney tubule. This is the normal tubular epithelium. You can see this cells. These are perfectly fine. However, this cells are swollen and these are filled with water. So this is called as hydropic change. Besides that, the calcium influx because of membrane damage, particularly in the mitochondria, that will again cause swelling and deposition of phospholipid-rich amorphous densities. Now, this amorphous densities we cannot see in light microscope. For this, we need electron microscope. Now, other changes which can be a part of reversible injury in fatty organs, we can see fatty change. We can see myxoid change, like this mucinous material is deposited in the matrix, or we can see some deposition of hyaline material. So these are all changes seen in a reversible injury. Now, what will happen in case of reduced protein synthesis? Now, when there's continued hypoxia, again, the membranes of endoplasmic reticulum and Golgi apparatus will swell up. There will be detachment of ribosomes, and this dispersed ribosomes will get dispersed in cytoplasm, and the function will be inactivated. Now, what happens because of this decreased protein synthesis or decreased RNA synthesis? There is increased eosinophilia. As you can see, this cytoplasm is not that pink, while in case of this in this cell, the cytoplasm is appearing more pink. This is called as hypereosinophilic state, or there is increased eosinophilia. This occurs because this stain, can anyone tell me what is this stain? You must have seen in your normal histology class also. Yeah, this is hematoxylin and eosin. So usually the hematoxylin gives a blue color. So this nucleus appears blue because of hematoxylin, while the cytoplasm takes up eosin. That's why it appears pink. So what happens? The ribonucleic acid that has affliction for the hematoxylin in a normal cell. When there is decreased amount of ribonucleic acid, the hematoxylin cannot bind, and obviously it will appear more eosinophilic. So that is the reason for increased eosinophilia. Now, what are the ultrastructural changes? Ultrastructural changes are changes which we see in electron microscopy and not in routine microscopy. I have said about the plasma membrane alterations such as blebbing. So this, you can see, these are the normal microvilli, while this is the formation, the microvilli are completely blunted here, and there's formation of this blebs. So this is a part of reversible injury. Then this mitochondria, these are also appearing swollen up. This, you can see, this is the normal mitochondria, while these are appearing swollen up and rarified, and these are the amorphous densities. These amorphous densities can be seen in both reversible and irreversible injury, but it is more in case of irreversible injury. In reversible injury, the amount is not that much. And apart from that, we can see this accumulation of myelin figures. These are the myelin figures, which are what? These are basically aggregates of phospholipids, and these are damaged. These are derived from the damaged plasma membrane. Besides that, we can also see nuclear alteration like disaggregation of granular and fibrillar elements. So to summarize the reversible injury, if there is ischemia, there will be decreased oxygen supply. This will lead to decreased oxidative phosphorylation, decreased ATP synthesis, which will lead to inactivity of the sodium potassium pump. Because of this, there will be influx of calcium and water, and so this will lead to cell swelling, loss of microvilli, blebs, endoplasmic reticulum, and myelin figures. What are the other aspects? There will be increased amount of anaerobic glycolysis, which will lead to accumulation of lactic acidosis. So there will be clumping of nuclear chromatin and detachment of ribosomes from endoplasmic reticulum. and the other effect is decreased protein synthesis. Now coming to the irreversible injury. Up to that point, the cell was reversible, means it can be reversed back to the normal state. Now, if there is continuous insult, continuous decreased oxygen supply, continuous ischemia, the cell will move towards the state of irreversibility. So what are the two essential aspects of an irreversible injury? One is mitochondrial dysfunction, and the another is disturbance in the cell membrane and particularly in the plasma membrane. So what all damage will be seen? There'll be rupture in a reversible injury. We had seen that there is swelling of the organelles. In case of irreversible injury, there'll be rupture of the membranes. The lysosomal membranes will rupture, and it will release various enzymes, which will lead to, in turn, lead to damage of the cellular constituents. The endoplasmic reticulum, that will also show lysis. And also the main change will be seen in the nucleus in the form of pycnosis, karyolysis or karyorexis. I'll be discussing next. So what happens in irreversible injury when the insult is continuing? There is activation of the enzymes now. The phospholipase is activated, which will lead to phospholipid degradation. There will be increased phospholipid degradation. This will ultimately lead to membrane damage. There will be lysis of all the membranes. There will be protease activation, so it will also lead to cytoskeletal damage. And this increased amount of calcium uptake in mitochondria, this will lead to large flocculent amorphous densities. In reversible injury, there will be a small amount. In case of irreversible injury, we can see large amount of deposits. And also this release of this lysosomal enzymes that will cause the nuclear changes like the karyolysis, karyorexis and pycnosis. And also there will be decreased basophilia, means the cell's cytoplasm will be appearing more hypereosinophilic. Now this aspect of irreversible injury that can be also used in case of detection of diseases, like when these enzymes are released from a damaged cell, we can predict that which organ is involved. Like in liver, there'll be elevated aspartate aminotransferase or SGPT. In case of myocardial infarction of skeletal muscle injury, we will see there is elevation of CKMB. In pancreatitis, we can see amylase. So because of this cell damage, there will be release of enzymes into circulation, and then we can predict what is the progression of disease, how much damage is there from these enzymes. Now coming to the nuclear changes a bit, what we understand by the term karyolysis, pycnosis and karyorexis. In karyolysis, now there is decreased basophilia of the chromatin. The nuclear chromatin we know is basophilic. It appears blue in appearance. So there is decreased basophilia because of the disintegration of the DNA. And this is because of the enzymatic degradation by the endonuclease, which are released by the lysosomes. So the nucleus appears faded. Like here we can hardly see the nucleus here. The other thing is pycnosis. In pycnosis, what we see, there is nuclear shrinkage and increased basophilia. The chromatin then condenses into a solid shrunken basophilic mass. Like we can see here, as compared to a normal hepatocyte, here this is the normal nucleus. This nucleus is appearing much more shrunken. This is called as nuclear pycnosis. The third thing is karyorexis. Now after pycnosis, the nucleus will get disintegrated. That is called as the karyorexis or nuclear fragmentation. Again, what we will see in electron microscopy, in the reversible injury, we had seen the organelles appear swollen up. In case of irreversible injury, there will be discontinuities in the plasma membrane and the organelle membrane. There will be marked dilatation of the mitochondria, and there will be large amorphous densities, not tiny densities. The amount of myelin figures will be obviously more in case of irreversible injury. So once again, to discuss the mitochondrial changes in reversible injury, there will be cell swelling and amorphous densities. But in case of irreversible injury, the deposits will appear more dense because of increased amount of calcium accumulation, and also there will be increased there'll be appearance of vacuoles in the mitochondria. And in case of nucleus, while in reversible injury, there was clumped chromatin. In case of irreversible injury, we can see either pycnosis, karyorrhexis, or karyolysis. Now coming to the difference between reversible and irreversible injury. Now what are the biochemical alterations? There is decreased generation of ATP in both. However, in reversible injury, the amount is not that much. There will be loss of iron and fluid homeostasis. There is defect in protein synthesis, and there will be cytoskeletal and DNA damage. While in case of irreversible injury, there will be profound membrane dysfunction. There will be denaturation of the intracellular proteins. There will be enzymatic digestion of the organelles, and there will be breakdown. Means everything will be breaking down in irreversible injury. We can hardly see any cell outline in case of irreversible injury, which we see in necrosis. Now, what are the ultrastructural changes? There will be blebbing of plasma membrane, organelle swelling, detachment of ribosomes, and clumping of the nuclear chromatin. While in case of irreversible injury, there will be discontinuities. There will be severe mitochondrial damage in the form of increased amount of densities and dilatation, increased amount of intracellular myelin figures, and increased amount of denatured proteins. What we will see in light microscopy, while in case of reversible injury, we see hydropic swelling, then fatty change or increased eosinophilia. In irreversible injury, the eosinophilia will be more pronounced. There will be glassy appearance because of dissolution of all the membranes. Also, there we can see calcification and nuclear changes.